Hello everyone, I'm Ezenma Chukwuemeka from Group 182A, and I'm here to discuss about the biochemistry of muscle, connective and nerve tissues. We'll start by types of muscle tissues. There are three types of muscle tissues. We have the skeletal muscle, the cardiac muscle, and smooth muscle. The skeletal muscle has striated tubular, multinucleated fibers and is usually attached to skeleton. It is voluntary. The cardiac muscle. The cardiac muscle has striated, branched, uninucleated fibers, occur in walls of heart, and is involuntary. The smooth muscle. The smooth muscle has spindle-shaped, non-striated, uninucleated fibers, occur in the walls of internal organs. It's involuntary. Mechanism of muscle contraction. When activated, myosin heads attach to binding site on thin filaments and sliding begins. Each cross bridge attaches and detaches, creating tension that pulls the thin filament towards the center of sarcomere. This leads to cell shortening or contraction. Looking at the diagram here, this A part is showing the relaxed sarcomere, whereas this B part is showing the fully contracted sarcomere, showing the disappearance of the H zone and the Z disc which comes closer, and the I band which is nearly gone. Connective tissue. The connective tissue consists of two basic elements, the cell and extracellular matrix. And the extracellular matrix comprises of the fibers and ground substances which include the liquid, the gel or solid. Functions of connective tissues include bind or support other tissues. Component of extracellular matrix. It includes the glycosamine glycans and the proteoglycans, structural protein and adhesive protein. Collagen. Collagen is the most abundant protein in the human body. A typical collagen molecule is long, rigid structure in which three polypeptide alpha chains are wound around one another in a rope-like trough-like helix. The collagen fiber orientation: three fibrous layers creating a widely meshed network. Outer oblique fibers, inner longitudinal fibers. Fiber oriented 120 degrees apart. Elastic fibers run parallel collagen fiber within each layer. Proteoglycans. Proteoglycans, all of the glycosamine except hyaluronic acid and heparin, are found covalently attached to protein, forming proteoglycan monomers. A proteoglycan monomer found in cartilage consists of a core protein, as you can see here, which is brown in color, and covalently attached glycosamine glycans, which is blue in color as labeled here. And here is the basic structure of proteoglycan. Now the nervous tissue. The nervous tissue, the structure comprises of the nerve cells, which are neurons with cytoplasmic extensions, the neuroglial cells, which are supporting neurons. Their functions include sensing changes in the environment and respond by transmitting impulses. Location where these nerve tissues these nerves can be found include the brain, the spinal cord, and the peripheral nerves. The nerve impulse. A nerve impulse is an electrical message that is transmitted down the axon of a neuron. The impulse is regenerated at points along the axon. The speed of the nerve impulse ranges from approximately 1 milliseconds to 100 milliseconds. Now looking at the generation and conduction of nerve impulse. In contrast, the fluid outside the axon contains a low concentration of potassium, a high concentration of sodium, and thus form a concentration gradient. These ionic gradients across the resting membrane are maintained by the active transport of ion by the sodium-potassium pump, which transports 3 sodium outward and 2 potassium into the cell. Action potential. Action potential is all about the movement of ion in and out of the cell. At rest, the neuron has a resting membrane potential of -70 millivolts, which is negative on inside of the cell. When a nerve becomes excited as a result of a stimulus, the gated ion channel on the membrane open and allow the sodium to rush into the membrane. This is facilitated diffusion. No energy is required. Remember that the concentration gradient was established by the sodium-potassium pump. The sodium rushes through the membrane, causes the inside of the neuron to become positive. The neuron is now depolarized. Here is a graph representation of the action potential. Here is our resting membrane potential, assumed to be at -70 millivolts. When being stimulated on getting to the threshold, which is -55, there will be action potential which will cause depolarization, and getting to overshoot, which is the reverse of polarity, there will be the potassium gated channel is going to open and there will be efflux of potassium, which will be to polarization and then to hyperpolarization, then return back to resting membrane potential. Thank you very much for your attention and keep staying safe.